let's talk about something that is, without a doubt, a cornerstone of modern biology: building DNA from scratch. I mean, literally creating custom DNA molecules letter by letter in a lab. This is the chemistry that makes things like CRISPR, rapid disease testing, and even RNA vaccines possible. So, have you ever really stopped and wondered how that actually works? How do you take these basic chemical building blocks and assemble them into a precise genetic code that you literally designed on a computer? Well, that's exactly what we're going to break down today. Okay, so there's a fancy name for this, of course: oligonucleotide synthesis. Oligo just means short. So it's really just the process of chemically building these short strands of DNA or RNA, usually up to about 200 bases long, in any sequence you can dream up. All right, so here's our game plan. First, we're going to look at why this is so tricky in the first place. Then we'll dive into the brilliant two-part strategy that makes it all possible. After that, we'll spend most of our time on the four-step cycle that actually builds the DNA chain. And finally, we'll see how it all gets cleaned up at the end. So let's start with the big problem. The building blocks of DNA are super reactive. I mean, they're just itching to connect with things. So getting them to link up one at a time in the exact order you want without creating a total mess of side reactions, well, that is a huge chemical headache. To get around this, chemists came up with two incredibly clever ideas that are really the foundation of this whole thing. One is a way to control the chaos, and the other is a way to protect the molecules until the exact right moment. The first big idea is something called solid phase synthesis. See, if you just tossed all your DNA building blocks into a beaker, you'd get chemical chaos. They'd react all over the place. But what if you anchor the very first molecule to a solid surface? Suddenly, you have control. You can add your next ingredient, let it react, wash it away, and then add the next one. It brings order to the entire process. And this is what that anchor actually looks like. The whole process kicks off with a single DNA base, our first letter, stuck to a tiny, porous glass bead. This little bead is our solid support, our foundation. It's what we're going to build the entire DNA strand on, one piece at a time. Now for the second stroke of genius: protecting groups. Each new building block, it's a modified nucleotide called a phosphoramidite, comes with these chemical safety caps on all its reactive parts. The most important one is this big guy, the DMT group. It sits on the end of the molecule, basically telling it, nope, you can't react just yet. And we're not just talking about one safety cap here. Every part of the nucleotide that could possibly react is carefully covered. As you can see, the DMT group blocks the end where the chain grows, but other groups protect the actual DNA letters, the A, C, and G. And even the phosphate part is protected. It's all about making sure only one specific reaction happens at a time, exactly when and where we want it to. Okay, so let's recap. Our starting block is anchored down, and all our new building blocks are wearing their little safety caps. Now we can finally get to the heart of it all: the automated four-step cycle that adds one new letter of DNA with every single loop. This flowchart is basically our map for the next few minutes. A machine just repeats these four steps: deprotection, coupling, capping, and oxidation, over and over and over again. One full cycle for every single letter you want to add. So let's walk through them. First up is step 1, deprotection. To add a new link to our chain, we have to create an open spot for it to connect to, right? So the very first thing we do is pop off that main DMT safety cap from the nucleotide that's anchored to our glass bead. So here's the chemistry. We wash a mild acid over the bead, and it's specially designed to just clip off that one DMT protecting group. It gets washed away, and voila, we have a free reactive connection point just waiting for the next building block to arrive. And now for the main event, step 2, coupling. With that connection point wide open, we introduce the next protected building block in our sequence. This is the moment our DNA chain actually gets longer. So here's a crucial point. The new building block is first activated by another chemical called tetrazole. This makes it super reactive, so it immediately snaps onto that open spot on our growing chain. This forms a new bond, but it's a temporary, kind of unstable link. We're going to make it permanent in just a minute. But hold on. Before we make that new bond permanent, we do something that is just an absolutely brilliant bit of quality control. It's called capping. And this step is what ensures our final DNA is super accurate. You see, the coupling reaction isn't 100% perfect. A small fraction of the chains might fail to grab the new nucleotide. So what do we do with those failures? We permanently block them off with a different kind of cap. This effectively takes them out of the game. making sure that only the chains that grew correctly are allowed to keep going. It's genius. And that brings us to the last step in the cycle, oxidation. We've got that new unstable bond we just made, and now it's time to convert it into the strong, stable backbone that you find in actual natural DNA. To do this, we use a solution with iodine. The iodine basically rearranges the atoms and oxidizes that fragile link, turning it into a rock-solid phosphodiester bond. This is the exact same chemical backbone that gives real DNA its incredible stability. That new connection is now locked in for good. And that's one cycle done, one letter added. So to add the next one, you guessed it, the machine just starts right back at step 1, taking the safety cap off the nucleotide we just added, and runs through the whole 4-step process again. If you want a 50-letter piece of DNA, this cycle just repeats 49 times. Okay, on to the final stage. Once the very last cycle is finished, our full-length DNA is built. But it's still stuck to that glass bead, and it's still covered in all those other little protecting groups. So now we have to set it free and clean it up. And this is the great unveiling. A chemical, usually ammonia, is washed over everything. It has 2 jobs at once. It snips the finished DNA strand right off the glass bead, and it strips away all those remaining little safety caps from the DNA bases. We go from that complex protected molecule on the left to the clean, final piece of DNA on the right. So just to sum up those final steps, first, cleavage, cutting the DNA from the bead. Then, deprotection, taking off all the leftover chemical caps. And finally, purification. The whole mixture is run through a system that separates our perfect, full-length DNA from any of those capped-off failure sequences, leaving us with a pure, high-quality final product. And there you have it. That's how you build DNA from the ground up. It's an incredible process that literally gives scientists the power to write the code of life, paving the way for everything from new medicines to storing data in DNA. It really just leaves you with one fascinating question, doesn't it? Now that we can write DNA with this kind of precision, what should we write next?